Sounds like you've heard quite a bit already about the model. So I'll just kind of jump right in. So just really briefly, since you've probably seen a lot of this before, just some overview and motivation. So E3SM is the Energy Exascale Earth System model, which branched off from the Community Earth System model about 10 years ago now. And ELM is a land surface model component of that, which has its origin in CLM. So it does biogeochemistry, carbon, nitrogen, and phosphorus cycling, hydrology, energy balance. More specifically, we have 10 soil layers in the model that track moisture, temperature, carbon, nitrogen, and phosphorus, as well as tracking sort of the plant pools, live plant pools of these things as well. E3SM on the left hand, you just kind of see a picture of how the different components of the model are connected. Just a reminder that it is a coupled model, and land is one component of that, and it interacts through a coupler with an atmosphere, an ocean, and ice models as well. So land model uncertainty has been a big story for decades now, and even after lots of research, land surface model uncertainty remains large, especially regarding the carbon cycle, which you can see here on the right, sort of a kind of old picture now from Friedlandstein et al. In 2014, showing multiple models, modeling groups, and their predictions of cumulative land flux over the 21st century. And so that's how uncertainty has been diagnosed a lot in the past, is looking at just multiple ensemble members of different models. And these models tend to be quite expensive to run. But on the other hand, there's a need for ensembles. So running these models lots of times so that we can do uncertainty quantification, or I'll refer to that as UQ. And that includes things like global sensitivity analysis, where we try to understand which uncertain model parameters are really driving uncertainty or variability in model outputs, and model calibration, where we bring in observations or benchmarks to try to tune or calibrate those parameters to their optimal values and reproduce the data better. Of course, as I already mentioned, some major challenges. These models are quite expensive. This is sort of changing now, but in the past, modeling groups have only been able to complete a few ensemble members. That's growing now as computational power grows, but we still have this problem where we have lots and lots of outputs, and those outputs are very dense in space and time. And so we look at sort of the total number of variables that are produced from these runs. It's a very large number. So there are some needs, which I'll talk a little bit more about how we're addressing that, sort of in a way to reduce the dimensionality of these huge outputs, and to construct surrogate models in which we don't have to run necessarily the entire model. Once we have an ensemble and some understanding of model behavior, we can develop a model of that model to sort of predict how the model would respond within that parameter space, even if we didn't evaluate the model at a particular combination of parameter values. And so just a little bit more about a little bit more motivation even. This figure compares sort of the ocean and the land uncertainty on the right here. And if you look at multiple models, multiple modeling groups, I think 12 here, over 4 different scenarios in which things like anthropogenic emissions are changing, we can see that over time, the ocean uncertainty is dominated by scenarios, right? So basically how much emissions are there. Whereas on the land side, this model uncertainty remains very dominant throughout the entire prediction period. And it's really the sort of structural and parametric uncertainty among the models that really drive the uncertainty in the predictions. So I think you've heard a little bit, at least, about the offline land model testbed, which is kind of running under the hood and some of the exercises that you're doing now. And so the objective of the OLMT, as I call it, is to simplify the workflow. And if you look at sort of what you have to do in order to run sort of even a single site simulation, it's quite complicated in terms of the number of steps that are involved. And so, for example, we have to do a full site simulation with the biogeochemistry enabled. We typically perform 2 phases of spin up. So an accelerated decomposition spin up, in which soil and mortality pools are accelerated, and then sort of transition that into a regular spin up period. at the end of which we have kind of a steady state pre-industrial carbon uh, carbon cycle, and then we begin a transient simulation, typically in 1850, where we begin to apply rising CO2 concentrations, nitrogen deposition, land use change, and meteorology. And so it's typically kind of at the end of that transient period where we actually have data from towers or other sources that we want to compare the model against. But in a total, that can involve about 1,000 years typically of model simulation. And then in order for each one of these cases, as we call it, there are a number of steps involved, creating the case, setting it up, compiling the code. In the case where we want to do ensembles or test multiple parameter values, we've got to manage all of those simulations that are running potentially in parallel, and then to post-process and sort of get the outputs that we care about. The model, I think, generates over 500 output variables now. Typically, we want to just look at a few and sort of aggregate all these output files and look at temporal behavior at a given site. So we can take as input, we can run either a single set of parameter values, or we can give OLMT a range of uncertain parameters. And so for each parameters, and this is just an example of an ensemble that we might want to run, so some parameters that we might want to vary, we can give the code minimum and maximum ranges for each of those, and then we'll use that information to construct an ensemble of some given size, say, for example, 100 or 1,000 ensemble members, and manage those for you, and then give you the outputs for each one of those simulations. And so what do we want to do with that? So one of the things I wanted to talk a bit about here is surrogate modeling. So a surrogate model, or emulator, as some folks refer to it, in this context is a model of ELM's response of an output variable or multiple output variables to parameter changes. And so, for example, we might have 1,000 ensemble members that we've run using the OLMT framework, varying some number of parameters that we can then take, and we can fit, for example, a neural network to that in order to have a surrogate model that can predict the model's response. So, for example, here NPP, annual NPP at one site. As a function of those same parameters, but will allow you to predict what that NPP would be at a point in that parameter space where we didn't actually run ELM. And so this is a very high dimensional space given the number of parameters that we're looking at. So I think it's very valuable to have that ability to predict what the model would do at a new point. And so there are multiple ways to create these types of surrogate models. What I'm showing here is just an artificial neural network. Other machine learning techniques could be used, and some are better in certain situations. The neural networks tend to work pretty well. And as part of this process, part of this training process, a lot of times what we need to do is hyperparameter tuning. For example, in a neural network, that could involve the hidden layer number, size, activation functions, solvers, or learning rates. With other methods, you might have different hyperparameters. But given that, if you have that ability to tune the hyperparameters, typically you can come up with pretty good fits of these surrogate models. And so in this example on the right, for NPP, we're getting quite a good R squared of about 0.98, and some other variables shown here below. So this becomes quite expensive, though, when we have lots of output variables. And so here it's just NPP for 1 year, but we might be interested in a daily time series over many years, or a gridded simulation where we have lots of grid cells that we want to predict, or both space and time. So that number of outputs then quickly can jump into the thousands or even millions. And so that's where we start to bring in dimension reduction approaches to train a surrogate in a reduced dimensional space, rather than having to do these sort of training exercises millions of times. And so here's just a little bit more detail on the workflow for automated calibration. And so we might have these same parameters coming in. We're running these ELM ensembles, generating a surrogate model. What we can do then is take that output variable, compare it with observations, and get a set of optimized parameters that we can then feed back into the real ELM model, not its surrogate, to then sort of update the parameter file that we might want to use in future simulations. And one of the things that can help with is sort of knowing that optimal set of parameters, so the parameters that best fit the data, can reveal sort of times when the model still isn't working very well. It might give you some clues as to what part of the model structure still needs work or development. So here's an example of global sensitivity analysis. And so this is one of the things that's enabled by having these surrogate models. These analyses, Sobol sensitivity analyses, generally require a very large number of samples from the model, in which we might want to calculate main or joint sensitivity indices for model parameters. This here is an example from the Spruce experiment site that's published in this paper listed here. And so within these experimental enclosures, we have multiple plant functional types. We have experimental treatments, and so we really wanted to understand sort of what parameters are driving the variability in model outputs. And so, for example, on the left, we can see GPP and what parameters. So the relative height of the bars here are the higher the bar, the more sensitive that particular parameter is. And so, for example, this one leaf carbon nitrogen ratio of the spruce trees has the largest impact on GPP, followed by FLNR, which is the fraction of leaf nitrogen in rubisco. But we can repeat these for different problems, and we tend to see sometimes the same parameters coming out, but sometimes very different depending on what time what the temporal scale of the output is, and where the site is. These sensitivities can vary quite a bit. So one reason why we like to do these early in the process of quantifying uncertainty is it allows for down selection of parameters that we might then want to go and target for calibration. And so when we construct these surrogate models, we can get away with a little bit less accuracy to get reasonable or reliable sensitivity numbers. But to perform model calibration, typically we need really accurate surrogate models to sort of limit that potential error. So here's just a brief example of calibration. And so to do calibration, we're using a technique called Markov chain Monte Carlo. This is a Bayesian calibration method that requires typically lots of evaluations. So in addition to providing the sort of posterior maximum or best set of parameters, it also gives you confidence intervals around both the parameters and the outputs, which can be very useful as well. But it requires a lot of evaluations, and importantly, those evaluations have to be done in serial because it's kind of a chain. So 10,000 to a million serial evaluations for a model like ELM, where even for a single site, it can take multiple hours. You can imagine that exercise would take years. But in this case, we can save the outputs of interest from our ensemble. For example, about a thousand ensemble members, and importantly, those ensemble members can be run in parallel, right? So we can take advantage of clusters or big machines to run a bunch of these simulations at the same time, and then use the outputs from those to train a surrogate model, which might take just a few minutes, and then to evaluate the surrogate model within our Markov chain Monte Carlo chain can be orders of magnitude faster than running the full model. And so that's one of the big advantages of this methodology. And so the inputs to that surrogate model are the same parameters that we use in ELM. So for example, this FLNR parameter or this ball-bearies to model slope. And then we can calibrate that using FluxNet observations, for example. And so on the bottom is an example of an output that we get for a given parameter. So the FLNR parameter and this sort of PDF or probability density function for that parameter that we obtain after running MCMC. And so we get, I think in this case, we calibrated 10 parameters. So we have 10 of these. I'm just showing the most sensitive one here that we could then take back and use in ELM. And on the top is the observations in blue with their uncertainties, and then the model best fit in red, and then the confidence interval of model predictions, posterior confidence intervals shown by the dashed black lines. So as I mentioned, when we're running gridded simulations, or if we want to look at, long time series, reducing that dimensionality of our surrogate model is really important. And so we're using a method called Carhun-Love decomposition and combining that with our neural network methodology to generate the surrogates. So basically, instead of generating tens of thousands or more surrogate models, we might be able to reduce that dimensionality down to 10 or 20 and build surrogates for those, basically coefficients in this reduced dimensional space. So what I'm going to show here, some results from a paper that my colleague Kachik Sargsyan is working on. It's a methodological evaluation at 96 FluxNet sites. And so he was able to generate this kind of reduced dimension surrogate model and apply that to these sites and do things like sensitivity analysis and calibration. And so we can look at, so for example, over these 96 sites, these parameter sensitivities and how they vary across the different sites. And we can see here that this blue parameter, or FLNR, is almost always the dominant parameter in terms of sensitivity. The reason for that is it's basically a strong control on photosynthesis. It regulates VC and so it's a strong control on GPP, which many things will follow from that in terms of the land carbon cycle. We also see other parameters jumping out, some more at more sites than others. This MBB opt is the ball-berry stomatal conductance slope. We have VC SE, which is a parameter that affects the temperature response of VC the ball-berry intercept term would be the red one. And then I think this green one on top is the day length scaling factor that's used in ELM to reduce VC max when day length is small. So this, I don't know if it's showing the animation, is a comparison on the left of the actual results, GPP outputs from ELM in June, July, and August, and the surrogate model representation of that for June, July, and August for GPP. And so you can see the patterns are very similar. The surrogate model does a very good job at representing these different ELM model samples. And by samples, I mean for each sample, a different combination of model parameters was used to generate these outputs. So it's able to recreate the spatial patterns and also the variation caused by the parameters themselves. The next figure here is a map of sensitivity. So this, I picked the ball-berry slope parameter just to illustrate what that sensitivity looks like across space over the globe, and on the left in the winter, northern hemisphere winter, and on the right, the June, July, August period. So we can see ranging from almost no sensitivity to as high as about 0.5, which would indicate that about half the variation in the output is caused by this parameter. It seems this parameter kind of jumps out more in areas that are prone to drought, to where the regulation of water matters more. This parameter tends to jump out as a more sensitive one. And so also we can run calibration across all of these sites. And so what this plot is showing, sort of when we run these site by site level calibration, what the sort of the error in terms of relative mean squared error looks like, with the nominal parameters, kind of the default models that are used. And then if you looked at the mean of the ensemble that we ran of ELM parameters, it'd be the red. And then after calibration, where we actually use the data, to fit the best model parameters, we can see how much the relative error is reduced in GPP there. Of course, at some sites, quite a bit of error still remains, which would indicate that there's probably some structural issues with the model. It's not able to reproduce the data very well. But with many sites, we do get that relative error down by quite a large fraction of what it was with the default parameters, especially for some of these sites out here on the end. I think these are, can't remember if these are arranged by plant functional type. So these are more kind of like the tundra and grassland sites out here on the end. We can also see what those numbers look like in terms of this most sensitive parameter. That's the FLNR. These are color coded by plant functional types. And so some plant functional types, we have a lot more sites than others just due to the distribution of the towers. And we can see the prior range that we used here is the black bar. So we allowed values, I think, from 0.2 all the way up to 0.25. And then these smaller bars indicate the posterior uncertainty ranges, which I think are the 95% confidence intervals. So one of the things we wanted to look at here is how similar were sites of the same PFT versus across different PFTs. And so this blue one here, I believe, is temperate evergreen forest. So for example, we can see they tend to cluster around a certain value, but there is a lot of site-to-site variation of the optimal FLNR parameter. And that the same pattern is true for a lot of the other plant functional types as well. So I think this is useful to look at, and it may be that the PFT maps that we're using, there may be some cases where we might want to consider using more PFTs or clustering them in some different way where we can actually use sites that have similar optimal parameter values. So the next slide just shows a couple of sites more specifically. So the USMOZ, or the Missouri Ozark flux site, is on the top. The red sort of envelope indicates the model prior. And so when we feed these sort of prior ranges of parameters into the model for this site, you really can get a very large range of GPP. And these are seasonal cycles that we're seeing over about a 10-year period, I believe, or a little bit more than that. And then the green shows the posterior. So after calibration, we reduce that initial uncertainty by quite a bit by calibrating with the data. And we tend to get pretty close to the observed values that are indicated by the black dots, at least for the Missouri site. If we look here at the Harvard site, we can see we don't do quite as good a job. So the model posterior tends to be a little bit lower than the observations. And so this is a case where, if the ranges of parameters that we're feeding into this ensemble, if none of those match the data well, or if that range, none of them are sort of covering numbers as high as the observed, then we're not able to get a good fit. And so that might be an indication that we need to extend the ranges of parameters that we're using for this given site. We looked then at two different calibration regimes, and this is now at the global scale using FluxCom GPP. And so two different ways of doing calibration. One, just one global surrogate and coming up with one value for the entire globe for FLNR. And if we wanted to do that, here's what the probability density function looks like for that parameter. And then on the right, we said, well, let's do it one grid cell at a time. So calculate the optimal FLNR for every single grid cell. And there's the map of what that looks like. And we can go back and compare that to see how it compares with the plant functional type distributions that we actually do use in ELM. So we can see some interesting spatial patterns begin to emerge when we do this. Some of the dry areas tend to have very high values. Boreal forests have lower values, I believe, than the model uses. So this was more of a methodological demonstration, given that using the FluxCom GPP, there's not of real information in there beyond what the sites are providing. So we did a very similar exercise using satellite solar induced fluorescence, or SIF, and calibrating the model with that. So we can see kind of what the calibration did here in terms of the prior mean GPP that's here on the upper left, which would be just the kind of ensemble mean over, in this case, I think it was about 200 simulations that we ran globally. And then importantly, on the right is the prior standard deviation. So can be quite high, especially over tropical areas. But after calibration, here on the bottom, the posterior mean GPP, um, after calibration, it's quite a bit higher, especially in tropical areas, maybe a little bit lower in boreal areas, but the posterior standard deviation on the bottom right is much reduced. And so by applying this data, calibrating using SIF, we're able to reduce the uncertainty quite a bit. And then we can look at what the sensitivities are as well. And so similar to what I showed before on the top, FLNR and MBB op, these are the dominant parameters. The model outputs are really sensitive to those, and then some of the more insensitive ones showing up at the bottom. And then here now we have plant functional type level parameter posteriors for on the top FLNR. So we can see the black dots are the model defaults that the ELM parameter file contains. And then the box plots kind of showing the mean and the range of those after calibration. So we can see significantly higher values for a lot of the plant functional types. And so, for example, tropical evergreen forests jumped up quite a bit, so that's responsible for the increase in the posterior GPP that you saw compared to the prior mean. That's true of a lot of the other forest types as well. For the other two parameters, they tended to be, they're not quite as sensitive. There's a little bit less information, and so the uncertainty ranges tend to be a bit higher, and we see the in most cases less deviation from where the default values were. So just to summarize here, so model uncertainty remains really large, I think, for predicting terrestrial responses and feedback. So there's, I think, a lot of work to do. Hopefully some of these methods can help to both help the sort of different modeling groups and their models agree more and to understand what's driving the differences in both in terms of model structure and model parameters. So ensemble and surrogate modeling techniques can enable that this kind of UQ to do that. I think that can be done from site to global scales. And we can improve model performance compared to benchmarks with like those in ILAM, which I think you heard about yesterday. And I hope that scripting frameworks like OLMT can greatly simplify these workflows, right? So if you have data, you have a site you want to compare, or even multiple sites, that it's easy to bring that in and to run these sorts of exercises and improve the model. So questions, and so yeah, before I stop talking, just want to show there on the left. So something that I have under development for OLMT, sort of a GUI interface that you can go in and you can configure things and launch sort of automatically from your local machine and run on a cluster, or run on the Docker. So I'm hoping to have that ready pretty soon. If you're interested, please let me know. If you have any other questions about anything else, please feel free to contact me. My email listed there. I just wanted to thank also the multiple projects that have contributed to a lot of this work, including E3SM, the Spruce SFA, and G-Arctic, and of course the U.S. Department of Energy, which funds all of this work. Thank you.
Awesome. Thanks so much, Dan. If you have a couple minutes for questions, we probably have some from the group for you. Yeah.
Sorry, can you say a little more about what that SIF parameter or variable is that you were calibrating to for the global model?
Yeah, so SIF is solar-induced fluorescence, which is It's just thought of as a good proxy for GPP. So we don't actually output SIF from ELM itself. So that's sort of SIF to GPP translation is done through another machine learning model to sort of make that translation. And so once we had that sort of SIF informed GPP, then we use that to calibrate directly ELM's GPP.
Thank you.
Hey Dan, thanks for the talk. I was wondering about the surrogate modeling. In your experience, what's the maximum number of parameters you can try to apply with that thing?
Yeah, so I think as I mentioned, so for sensitivity analysis, we can get away with a smaller number of ensembles for a larger number of parameters. And so for that, we've done, more than 50 or 60. And we tend to get at least reasonable looking sensitivities. But for calibration, it really depends on the behavior of those model outputs. So typically I use about 10 to 15, right? So if we can do that sensitivity analysis step first and sort of reduce that number down to a reasonable 10 to 15, we tend to get pretty good responses, good fits for the surrogate models. But it's something we always want to look at sort of as a first step, how accurate are these surrogate models before we actually run the calibration.
Hey Dan, thanks. So I kind of do have a question do you have to build an emulator or surrogate model for each of the variables that you are interested to benchmark and then do that kind of step by step, or there's a way to do I don't know, is there a way to kind of build one emulator and then try to do that parameter calibration for all the variables that you are interested?
Yeah, thanks. It's definitely possible to build one emulator, one surrogate model to produce multiple outputs, right? You can put a very large number of outputs into your neural network. So in OLMT, I think the way that it's designed, yeah, you can give it a list of model output variables and give it a time frequency that you want it to produce. And so it tends to work pretty well in practice up to about 100 or so. Beyond that, I think probably we'd want to consider reducing the dimensionality through the method I presented or some other method, right? That we want to, otherwise the training can become quite time intensive.
Hi there, thank you very much. I have one question regarding the surrogate models cost function. Do you need to have different cost functions for daily, monthly, yearly estimates, or you have just the one?
Cost functions for the calibration. Yeah, the way that I've set it up, typically I just have a single cost function. And so if we want, and it's something that you might want to customize, especially if we have multiple output variables at different time frequencies, there are different weighting schemes that you might want to experiment with. But right now, I'm just using if there's an observation, whatever time scale, that observation and its uncertainty go into the cost function just as is.
Okay, because in your project for the USHA1, you saw this model is underestimate the GPP for the monthly scale, right?
Yeah.
So do you need to have a seasonal and monthly cost function for that part, or only daily?
For that, I think everything was done at the monthly scale, right? So we post-processed the model outputs at the monthly scale, and then compare that to the monthly files from FluxNet, and yeah, did the fit at that time scale. It would be interesting though you could do it with the daily or annual and see how it changes.
Thank you.
And do you think it's important to consider the accumulation of some parameters, or if there's some way to do that?
Sorry, I had a hard time hearing. Can you repeat?
Maybe I can closer. So do you think it's important to consider some key parameter changes along the time? I mean, I don't know, maybe for some parameter, with times go on, they kind of have accumulation for plants, then they change the parameter. And I don't know if this should be considered, or do you think it's important?
Yeah, I guess yeah, it's a good question. Parameters could potentially change over time. I guess the hope is that if we have if we do suspect that's happening, that we'd want to encode that into the model, right? If rather than doing that sort of as a fitting exercise. But one thing that we can do is we can calibrate the model at different time periods, right? And then look at the optimal parameters over how those change over the different time periods. And that might give us an indication that we might need to incorporate that temporal behavior.
Thanks.
Hey Dan, thanks for the talk. So one question about when parameterizing specific parameters. What's the best way to know the range of up to, like what's the minimum, maximum values for each parameter?
Yeah, so there's often a lot of subjectivity that can go into that. So where we have data, we definitely want to use that data. So things like the TRI database, for example, can give us information about distributions of carbon nitrogen ratios over for a specific plant functional type. In the absence of information, I tend to just use a plus or minus some threshold like 25% or 50%, and go from there. But it can be dangerous because there's certain parameters that, we have to make sure we're not exceeding the physical bounds of what that parameter can be. Like if a parameter can't be more than one, and for example, that we definitely want to take those into account as constraints.
What was that TRI database you mentioned? TRI database?
Sorry, I couldn't hear.
She's asking about the TRI database. If you could talk a little bit about what that is.
Oh, yeah. So the TRI database sort of is a plant trait database, right? So people have gone out and made field measurements, are invited, encouraged to submit their data to this, and then they're sort of organized and tagged by. There's many different variables that are there, right? And it's a large database that modelers can go in and try to match those to specific parameters. So it's not always straightforward, like right, given the parameters we have in ELM and given things that are measured. Um, so there's a few papers that have explored using that database in more detail. I've only used it just a little bit here. It's definitely, I think, a good source of information for these sorts of activities.
Do you mind so in the tutorial right after your talk, they're going to be opening the parameter file and manually adjusting values and seeing how that changes output and then comparing to tower data. Do you mind briefly mentioning why that's, while useful for educational purposes, generally not best practice for parameter adjustments?
Yeah, I guess right. I mean, it really kind of depends on the parameter but typically, when we're making these adjustments, and a lot of folks do still kind of hand tune their models, but there are lots of different ways to get an answer that might match the observations well. And so this is why I think I like to encourage the approach where we look at parameters together, and try to understand what the sensitive parameters are sort of as a group, and then calibrate all of those together to sort of understand how these processes might interact. And so you could go in and you could change just FLNR, and you could get potentially a pretty good fit, but you might then, if you don't look at multiple variables or multiple outputs, you might make the model better in terms of GPP, but you might sort of break something in terms of energy flux. So that, as a word of caution, we like to bring in as many sources of data, as many parameters at once as we can.
Do you have any more questions for Dan? All right. Well, thank you again. That was a really great talk. We appreciate your time.
Thanks, Jim. Feel free to reach out. Yep. Thank you.